Welcome back to the Sans Channel podcast, where we discuss the coolest scientific papers and preprints of our day.
Great to be here.
If you're enjoying these deep dives into the latest science, please hit that like button and subscribe. It really helps us bring you more of this fascinating stuff.
Absolutely. Every click helps.
So today we're jumping into something that really gets to the heart of gene regulation.
It's all about transcription factors, or TFs, and this fundamental question of how reliably their binding to DNA predicts what they actually do to a gene.
Right. We're looking at a big study in yeast, Saccharomyces cerevisiae.
Yeah, a really comprehensive one. They use some pretty advanced techniques to map things out on a huge scale.
Okay, so the classic picture, the one you get in textbooks, is pretty simple.
Right. TF binds near a gene.
And boom, it flips a switch, on or off, like a light switch.
Exactly. Direct cause and effect. And, that model isn't wrong. We've seen it work for specific cases.
For sure, for individual examples.
But this study asked, does that simple model hold up systematically, like across almost all the TFs in yeast?
How did they check that? You mentioned big data sets.
Yeah, they combined info on where TFs bind using ChIP-seq, which gives really precise locations, with data on how gene expression actually changes using RNA-seq, so binding and function side by side.
So the key question, the thing that really grabs me is, does that TF sitting there guarantee it's controlling the gene? Is it always an active switch, or could it just be, hanging out, parked?
That's precisely the puzzle. And honestly, the main finding is, it's pretty surprising.
Oh, don't leave us hanging.
The overlap, between where a TF binds and which genes it actually regulates, it's much lower than you might expect.
Lower. Really? So like a TF binds to maybe hundreds of spots.
Or even thousands in some cases.
But it only actually regulates a fraction of the genes nearby.
Exactly. A significant chunk of those binding events don't seem to directly cause a change in gene activity, either up or down.
Wow. Okay, the paper says, our analysis reveals a limited overlap between TF binding and gene regulation. That really shakes things up.
It does. It challenges that simple switch model quite directly. Just because the TF is bound doesn't automatically mean it's functional at that location or at that time.
So let's talk about the binding itself. They use ChIP-seq to find all these spots.
Across basically the whole yeast genome for nearly all the TFs. It's a huge mapping effort.
And did they find any patterns in where they bind, like near the start of genes?
Yeah, actually, Figure 1A shows that lots of them, activators, repressors, the ones that do both, tend to cluster near the transcription start site, the TSS.
Which makes intuitive sense, right? You want to be near the starting line if you're going to regulate the race.
You'd think so. But again, proximity doesn't equal function, apparently. That low overlap hangs over everything.
Okay, what about the DNA sequence itself? TFs recognize specific patterns, motifs, right? Like a key fitting a lock.
They do. Each TF usually has a preferred DNA sequence it likes to bind to.
So did having the right motif at the binding site make regulation more likely? Did that explain things? I think figures 1C and 2A touched on this.
They look closely at that. And it's part of the story, but not the whole story.
Oh, okay.
Having the motif often means stronger binding, sure. But even strong binding at a perfect motif site didn't always translate into gene regulation. The paper actually mentioned something like only 6% of the TFs contain a significantly enriched motif in their set of function-only regulatory target genes.
Well, only 6%. That's tiny.
It is. And maybe another 7% have motifs in many targets, suggesting they might be, pioneer factors that open up DNA.
Right, the ones that get in first.
But for the vast majority, the motif alone isn't the deciding factor for regulation. Binding, even with the right code, isn't enough.
It's just getting complicated. So if it's not just binding and it's not just the motif, what else is going on? Context.
That seems to be the key takeaway. Context-dependent regulation.
Meaning
a TF's action probably depends heavily on other things happening around it.
Like what? Other proteins.
Exactly. Cofactors are a big one. Other proteins that might need to team up with the TF to get the job done. Like needing a specific tool or a partner.
Okay.
And also the chromatin environment. How tightly packed the DNA is. Can the TF even get access properly?
Yeah.
Can other machinery get in?
So it's like the whole neighborhood matters, not just the TF's house address.
Precisely. The paper notes that TFs often cooperate, and their binding sites are often clustered together.
Teamwork makes the dream work, even for proteins.
Kind of, yeah. They mentioned something called the sisal complex in yeast, hinting that TFs might hang out in these functional groups. cooperation might be more common than we thought.
Okay, and they also did these really cool experiments where they just deleted the TFs rapidly. Figure 4 shows this.
Yeah, rapid depletion. They used a system to essentially make a specific TF disappear very quickly from the cell.
And then watched what happened.
And then watch what happened to the expression of genes the TF was known to bind to.
Did everything go haywire?
Not necessarily. For many TFs, when they were suddenly removed, a lot of their supposed target genes, the ones they were bound to, didn't actually change expression much.
Oh. More evidence for that disconnect.
Exactly. Figure 4b gives examples like GCN4, CLN3, CIC8, Rev1. Removing them had really variable effects. Some bound genes responded strongly, others barely noticed the TF was gone.
That's pretty definitive, isn't it? Binding just isn't the same as regulating, at least not all the time.
It really drives that point home.
The study also tried to classify TFs, right? Activators, repressors, or both. Extended data figure 5.
They looked at whether a TF tended to turn genes on or off overall.
Were they neatly sorted?
Not really. Turns out most yeast TFs seem to be capable of both activating and repressing genes.
Dual function.
Yeah, depending on the specific gene, the conditions, probably those cofactors we mentioned, the same TF could push expression up or down and challenges the idea of rigidly defined roles.
So they're more like versatile tools than simple switches.
That seems like a better analogy, yeah. And even TFs that bind to tons of genes might only be actively regulating a smaller number at any given moment, and the effect could be activating here and repressing over there.
And just quickly, they double-check some of this with reporter assays. Like in figure 4a.
Yes, that was another piece. They took specific DNA bits where TFs bind, hooked them up to a reporter gene, and asked, does this piece of DNA with this TF binding site actually drive expression changes? A more direct test.
Got it. So, wrapping this all up, the big picture seems to be complexity.
Definitely. The simple idea that TF binding directly and reliably predicts gene regulation, well, it doesn't hold up across the board.
Binding is necessary, you can't regulate without it, but it's absolutely not sufficient.
Precisely. That limited overlap is the headline finding. Binding is just step one, and maybe not even the most important step in determining the final outcome for the gene.
Which leaves us with that big, fascinating question. What are all those other factors? Is it those cofactors showing up? Is it the way the DNA is folded? Is it timing, other signals in the cell telling TF, okay, now you act?
All of the above, potentially. It's clear that the decision to regulate a gene is, integrated from many different inputs, not just the presence of one TF.
It really paints a picture of a much more dynamic and interconnected regulatory network than just simple lines connecting TFs to genes. Lots to think about there.
For sure. It opens up so many avenues for figuring out how these decisions are actually made at the molecular level.
Absolutely fascinating stuff. Okay, that's our deep dive for today. Please don't forget to like the video and subscribe if you want more content like this.
Thanks for listening, everyone.